I think that with respect kind of the waves, if you will, in synthetic biology, initially the kind of first wave was focused on demonstrating proof of principle. So demonstrating things like toggle switches, demonstrating things like oscillators, where you can demonstrate that we can obtain control over cellular functions. And so seminal papers by Jim Collins and Michael Elowitz that at the time we also published on engineered cell-cell communication and being able to create basic devices, regulatory devices, and control their properties. And so I think those, around the year 2000 really set the stage for synthetic biology. And the second wave focused then on understanding how to take those basic capabilities, toggle switches, oscillators, multi-input logic functions, and begin to assemble them into larger systems. Where now, they're really beginning to look like sophisticated programs where there's multiple interactions between various kinds of modules that control one another, that provide input to one another, that feed back onto each other. And all of a sudden, this begins to look like a real programmable environment where you could do things that really were not imaginable before. Things where you can tell the cells to remember something, and based on what happened in the past and what's going on now, to maybe change their functionality, to have the ability to have multi-step programs that execute something. And then, one of the things that, for example, we're using that for is to control the development of organoids. So we start with basically stem cells, induced pluripotency stem cells, and we drive them to differentiate to liver organoids. And when the cells mature enough, we can then activate a second layer of differentiation and maturation. So that capability is really, I think, characterizing what the second wave was about. But we've now, I guess, hit this third wave where there's actually multiple aspects of the third wave. I think one aspect is the ability to take these functions and now demonstrate that they matter both in terms of industrial applications as well as clinical applications, where all of these modules, these safety switches, these multi-input logic functions that can really activate in specific cell types but not in other cells, I think are really making their way into the clinic. And I think they're going to result in significant improvements in efficacy and safety of various kinds of therapies, certainly for cancer, but also, vaccination, but also other kinds of, for example, autoimmune diseases and other kinds of application areas. So I think that the transition from the lab to the clinic is a really important one, but simultaneously, I think we're also gaining more respect for the underlying biology and really trying to incorporate more systems biology and a deeper level understanding of the biological systems in how we create synthetic circuits that operate properly, that operate in spite of cellular and environmental fluctuations. So I think the deeper understanding of biology is really changing the way we are now engineering systems. More, we appreciate more, and we take into account more of the things such as changes in cellular resources that are available to the circuits, and crosstalk interactions, multicellularity, heterogeneity, and so on. And I think also as part of that, we're also beginning to explore abstractions that are not just kind of pure digital binary abstractions, and trying to understand, are there different ways that we don't necessarily, use in other environments that are really well suited for the biological substrate. For example, one of the interesting ones that actually we are, in a sense, borrowing from computer science is the notion of neuromorphic computing. So programming cells using this notion of neuronal networks that were essentially neuronal networks machine learning, artificial intelligence, has been inspired by the way the brain works, and those have resulted in really a fundamental shift in the way computer programs for various kinds of applications, including pattern matching applications, in the way those programs are actually written. And what we're doing now is actually trying to bring it back to biology and understand how can we create genetic regulatory networks that operate like neuronal networks inside individual cells, but not using kind of neuronal interactions, but rather gene regulation. So I think that there's the third wave really consists of these different aspects of transition to the clinic, to industry, a greater appreciation of the underlying biology, and a move towards finding new abstractions that can work really well.